Of all the wonders of the world, none proves more miraculous than the human body. From tiny cells, muscles grow, flexible and strong. The mind develops, knowledge and wisdom increase. As children and then adults, we constantly strive to unravel the mysteries presented to us. We improve our quality of life. This is our goal, to improve the quality of life by fighting disease and improving health. We accomplish this by discovering and developing new and innovative medicines. It requires a large number of people from many disciplines working together with highly sophisticated technology and a wealth of scientific knowledge. Discovering a new medicine is an expensive and lengthy process. It can take as much as $500 million and up to 15 years to develop and deliver a single new medicine to the pharmacy shelves. The search begins with the selection of a disease or health problem. Many factors are considered, including unmet medical needs or any treatments currently available. Researchers must develop a detailed understanding of the disease, including the symptoms, affected organs, cells, and even specific cellular processes. The goal is to target a point where a drug might intervene to have the desired effect. This difficult task of identifying and understanding the fundamental process of disease may be aided by genetics. Recently, scientists have identified most of the over 100,000 genes in the human genome. Sophisticated analysis of this complex information, known as bioinformatics, may lead to increased understanding of the root causes of disease and to improved medicine. Researchers must start the search with a vast number of unique chemical and biological materials. Medicinal chemists in the laboratory produce many new chemical molecules for testing. Synthesis of each previously unknown molecule requires significant time and effort. Using a process known as combinatorial chemistry, robotics assist in the generation of large numbers of molecules. Biological materials such as purified enzymes, receptors, bacteria, and living cells are also required. Scientists use large-scale equipment known as biological reactors to provide extensive quantities of these materials. Using the new chemical molecules and biological materials, researchers begin their process of elimination. This advanced technology, known as high-throughput screening, allows scientists to test hundreds of thousands of molecules in a highly automated fashion. The process identifies the types of molecules on which the scientists should focus. Another way scientists find new treatments is through three-dimensional modeling. Once the three-dimensional structure of the disease target is known, scientists can look for chemical molecules that interact with it to have the desired effects. The process is similar to fitting a key into a lock. Only the precise key that fits the lock perfectly will work. This technology focuses efforts on a smaller number of molecules. However, scientists often do not have sufficient information about the target to use 3D modeling approaches. High-throughput screening and three-dimensional modeling can help scientists identify several molecules which become starting points for further testing. To allow for this testing, chemists must then make larger quantities of the selected molecules. To increase the chances of finding the best chemical molecule, they make additional molecules that are similar in structure. This starts the search for the ideal drug molecule. Once sufficient amounts have been made, the chemists work closely with biologists who conduct increasingly complex tests. They start with isolated cellular components, then move on to cells and tissues to determine how effective the molecules are against the selected disease. The physical and chemical characteristics must also be tested, such as solubility, or how well the drug dissolves in liquids, and stability, which refers to the ability of the drug to remain unchanged over time. The biological, physical, and chemical information collected to this point on the potential medicine is evaluated, and the best candidates are selected for further testing. This requires even larger quantities of the molecule. Synthesis of the molecule, also known as the active ingredient, progresses from laboratory scale to pilot plant scale. Formulation scientists work to convert the active ingredient into a dosage form that can be used by patients, such as a tablet, injection, or liquid. Sometimes, innovative methods must be developed for delivering the new active ingredient. The process for making the new dosage form must first be determined on a small scale in the laboratory, and then increased to produce the quantities needed for further research. Before more testing begins, it's critical to learn how the medicine reacts in a biological system. This involves extensive analysis to understand how the body breaks down the medicine so that byproducts may be studied. This also helps determine the appropriate dose levels. Once there's enough evidence to suggest the medicine is safe, clinical trials are ready to begin. Clinical trials are when the new medication is given to a select group of people. The results are strictly monitored and recorded to see exactly how safe and effective the new medicine will be. Information about the medicine and the plan for proposed clinical study is reviewed and approved by an independent review board charged with protecting the participants. The volunteers are given information about the trial, and their informed consent is obtained before they participate in the study. The first trials are with healthy volunteers to evaluate the safety of the medicine. The medicine is then tested in patient volunteers to evaluate its effectiveness on their specific condition. The medicine is evaluated in hundreds to thousands of people. The process to this point has typically taken 10 years. And if the medicine is shown to be safe and effective, the job is not finished. All medicines sold in the United States must be approved by the Food and Drug Administration. This requires the submission of research findings, the results of the clinical trials, and all the information gathered to this point. A typical application can be 60 to 80,000 pages. Once the information is delivered, personnel remain on call to answer any questions that may arise. Meanwhile, the processes for manufacturing the active ingredient and dosage form and for packaging the medicine are scaled up. Special measures are taken to make sure manufacturing has minimal impact on the environment. Teams of commercial experts are involved throughout the entire process of developing a new drug. They carry out many functions, from determining who the competitors are to searching out unmet medical needs. They also perform extensive marketplace research to determine what the ideal product would be for each new drug, including providing input on the ideal dosage form and its color, shape, and size. Once a drug is approved, representatives are trained to deliver detailed information about the new medicine to physicians and patient groups. A previously unknown molecule has now become a new medicine, helping meet the needs of patients from all walks of life. But the job is not finished. The safety and effectiveness of the new medicine continues to be studied after it's released to the public. This extensive effort involves having trained personnel in place to answer questions and receive customer responses. The discovery and development of a new medicine takes many years. It takes a diverse group of highly talented and relentlessly dedicated people to overcome the obstacles along the way. The challenge is difficult, and the odds against success high. But with every new medicine that is discovered, someone's life is made better. The science of discovery, improving the quality of life.